focus in and zoom in, and you can actually see, oh, there are several genes within this lost region or this deleted region, and what are those genes important for, and they're missing, so maybe some of those genes in there are required for proper diaphragm development. So then we go ahead and we look at all those different genes, and we test those and look at those. So that's what we're actually doing among a number of different patients. And we've recently run a new type of array. Even though we run it on everybody, we have a newer one that's very closely spaced with the genome, so you can actually look really close together and see what people are missing or not missing. And we've run that on patients who have more complex phenotypes, or they have more than one CDH and they have cleft lip and palate or limb anomalies or things like that, because we found this type of technique is quite helpful for the more complex cases. And we're actually analyzing our data now and validating it. So another type of, and if anyone has any questions, I'm sure it feels like, just ask, it's no big deal. We use another type of array, and this is called a SNP array. So that's what the technology now is using, all these different kinds of basically microchips, and we're putting lots of different things on them and run it. I mean, it's very technical, so if you have any technical questions, we can answer after or during. But on this type of array, we can look for different genetic variation. We can also start looking at disease susceptibility. And that's sort of the newer way that we're going into. And just to give you an idea of a typical array study, this is just sort of a generic description. Now the colors, the different colors on the two circles represent single nucleotide variations, or what Dr. Longoni was showing you, the AGCT variations. So you can compare two groups. So we're comparing the CDH group and the non-CDH group. And then you look at how frequent your nucleotide variants are in one group versus another. So that the nucleotide variants that are more frequent in the CDH group might be different from the ones in the non-CDH group. Like we see a lot of blue variants in CDH compared to non. So those blue variants might actually tell us, oh, we should go look in this particular chromosome or this region. Those genes might be more important for the CDH group versus the non, as being causative for CDH. So to give you sort of an example, we use those SNP arrays to look at large consanguineous families, meaning that they have blood-related parents, so the parents are related. And we used this particular SNP array to look at Denai-Barrow syndrome, which is a CDH-associated syndrome. Denai-Barrow syndrome is often seen with CDH, with absence of part of the brain, with a hernia in the belly button, with nearsightedness, with deafness, widely spaced eyes, with a really large soft spot when the baby's born, with nose and eye abnormalities. And we collaborated with a physician from the United Arab Emirates, and she sent us blood samples from a number of different family members in the United Arab Emirates who had Denai-Barrow syndrome, as well as from grandparents and all across this family tree here, which you can see. And actually, across many generations, we have cousins. So it's sort of, and then here, these are the affected individuals with Denai-Barrow syndrome. And these are two little kids who have Denai-Barrow syndrome. So what we did then was we said, oh, this is interesting. Let's use our SNP array, and we used homozygosity mapping, which I won't bore you with right now. But we found a region on chromosome 2, which here, this is the region, and all of these genes are contained within that region. So we decided to start sequencing different genes in that region, and we sequenced all the genes in red. And we eventually got to LRP2, or Meaghann, and we found out that was the gene responsible for Denai-Barrow syndrome. And actually Meaghann, or LRP2 is called, it makes a protein that actually regulates, it's a homeostasis protein that actually regulates certain vitamins and hormones and steroid hormones in the body. So without this special protein, a lot of the kids have, during embryology, they don't form, a lot of the organ systems don't form correctly. So it's not only, that's why you only don't see just CDH, you see a bunch of other issues with these kids. So this is the protein, this is the LRP2, or Meaghann, and you can see these are different mutations from different kids. We ended up actually sequencing we sequenced about, I think, 30 patients for Meaghann gene, and it's a huge gene. So it ends up being extremely time-consuming and expensive, unfortunately, because it's so big, and the kids, as you can see, have mutations all along the gene. So even having a mutation in a single gene is still complex. So it's not just you sequence this little spot, you know, a little snip and you know that's it, or a little spot and you're done. You have to sequence the whole gene for each child that you suspect has Mauro syndrome. So we get families from all over the world, or physicians calling us, asking us, you know, we think the child has the Mauro syndrome, would you sequence it? So that's why we've done so many kids, and now we're actually looking at ways to supplement these children with these hormones and things like that, because the disease is progressive. They still have eye issues. The kids who make it still have a lot of problems to deal with in their lives. So we're trying to help them after they're born to see what we can do as well, because we can test them and tell them they have this, but then we have to try to figure out what are we going to do after. So that's what we're doing with the Mauro syndrome now, is looking for therapeutics. So I mentioned a bunch of ways that we obtain data. So now we have all this data, basically, and we have to analyze it, we have to mine the data, we have to try to figure out what's going on, and then we have to integrate it. So we have biostatisticians at the Broad Institute and at MassGeneral who's now working to just sort of filter through and figure out what's going on. And that's what we're doing now as well, is networking gene networks, looking at pathways, and I'll stop here so you don't go nuts. But this is just sort of how we do things in our study and where we're going and the directions that we're heading in. But, and this is the last slide, but for the study to participate, I just want to give you some information, and we do have packets and stuff like that with us that can give you, and a bunch of you guys are already participating in the study, so you probably don't need to even go there, but I brought some of the newsletters, and stuff like that if you guys don't have any. The second newsletter, unfortunately, we only had 2 copies because we're in the process of ordering more. But they talk about different science that we've done and CDH and all different kinds of things, so feel free. Yeah, so the second one you can look at, but 2 people can have them, and that's about it, and I can send them to you too. So to participate, a family would fill out a study questionnaire. We perform a physical examination on the affected child. If not, we just ask for a copy of physical examination or from the hospital records, from when the baby, hospital delivery records or anything like that. We also collect blood or a pathology sample in order to get DNA from the affected individual, or as well as participating family members, usually the parents for sure, because we often check what we get in the child compared to the parents. That's pretty much what we have to do, especially when we find things on an array or something like that.